Good afternoon, Professor Periasamy. Thank you very much for being here and to have agreed to this interview regarding functional imaging and fluorescence lifetime imaging. Last month, Alessandro Esposito posted the most prolific authors on lifetime imaging, and you are one of the 5th best. So congratulations.
Thank you.
And would you mind to share with us some of your research in functional imaging?
Yeah, thank you very much for inviting me. It is always a great pleasure to be here, talk about the lifetime for functional imaging. I have done a lot of work on the research side of using the lifetime. Currently, I am working on fluorescence lifetime redox ratio using endogenous molecule to measure the metabolism and also mitochondrial dysfunction in live cells and in tissue and in live animal. That's my main research right now I'm working on.
So everything label free. So you are able to follow metabolism without any labeling or external fluorescence dye.
That's correct. It is NADH, FAD, and tryptophan provides autofluorescence. You do not have to label it. So this kind of technique is useful for translational research. More importantly, the lifetime is the technique possibly you can apply for this kind of measurements because it provides bound and free these coenzymes, NADH and FAD. It's only possible to discriminate these two functions, bound and free, using the lifetime technique. Intensity technique, it is impossible to do that.
Okay, thank you for the explanation. What do you think were the most relevant development in lifetime imaging in the last 15 years?
Well, so the lifetime imaging mode started in 1990s. Before that, the lifetime technique is available only in chemistry and physics lab. The important thing we have to think about the interpretation of the lifetime to the biological data. That is where the advancement the people are looking for, the biologists basically looking for how I can connect the lifetime measurements to the biology. So that requires some kind of education to educate them how the lifetime technique is maybe useful or for their biology.
So helping also in the interpretation of the data, where the molecular interaction that changed the lifetime, for example, is coming from.
That's correct. The lifetime technique is very well used in the case of molecular interaction, protein-protein interaction, which is called, we call it as a FLIM. So that is how the lifetime technique is advanced.
So there were a lot of developments in the last years. What are we still missing for the fluorescence lifetime imaging part?
So the still missing part in the lifetime measurement is the speed and also the data analysis. Because the biology is the nanosecond even. We can measure the lifetime in the nanosecond time mode, but the technology has to advance to speed up the data collection and also not to photo bleach. And in real time, if we display the data analysis part while collecting the lifetime, the biologist can easily interpret the results of the measurements.
So you think lifetime has still a lot of place in the biology lab and helping with the new technology will spread this new contrast in fluorescence microscopy?
That's correct. The lifetime imaging is a very good contrast technique. Not only contrast, and also you can do the dynamic imaging, even when the protein-protein molecule interacts. So this is a dynamic technique. It will advance pretty sure. It will be available in all biology lab or microscopy facility like a confocal. I think the FALCON system, I think in my opinion, the FALCON system satisfies that condition, but still it will, the biologist has to catch up what's going on. Yeah.
We should have some workshops soon.
Yeah.
Thank you very much. for your time. It was really a pleasure talking to you today.
Thank you very much for your time.